Hello students, welcome back to my channel. Today I am going to discuss the exercises of cell cycle, cell division and structure of chromosomes for class 10th ICSE, right? As well as CBSE also. First we go through the exercise here. Name the following. First, the shortest stage of mitosis is anaphase. Second, the structure which initiates cell division in animal cell, it is centriole. Third, see, the tip of the chromosome which are rounded and sealed, it is telomeres. Next, the type of chromosome whose centromere is in the middle, metacentric. Next E, the stage of mitosis during which nuclear membrane and nucleolus reappears, it is telophase. A membrane that disappears during late prophase, it is nuclear membrane. A pair of chromosomes carrying dissimilar alleles for a particular character, heterozygous, acts as barrier of hereditary unit, gene. Two animals which have 19 pairs of chromosomes, cat and sheets. A region of plant body where cell division occurs very actively. Cell division occurs at the tip of the root tip and the tip of the shoot tip. Growing parts, they occurs very rapidly. Structure that initiates cell division. That is in case of the animal cell structure which initiates cell division, it is centriole. Chromosomes appear thread-like, that is in the lactotine stage of prophase 1. They thread-like in the beginning of the prophase. The type of cell division which occurs in the cells of the reproductive organ. In the reproductive organ, the types of the cell division occurs, it is the meiosis, right? Now here, the exchange of chromatid parts between the maternal and paternal chromatids of a pair of homologous chromosomes during meiosis, it is zygotine, right? The number of chromosomes present in a nerve cell of a human being, naturally the same number as present, that is the 46 chromosomes or 23 pairs you can say. As here number of chromosomes, so it is 46. Now next, find the odd one out. Growth of a cell, synthesis of DNA, synapses and synthesis of protein. See, synthesis of DNA, synapses, synthesis of protein, they all occur in the cell division, whereas growth of a cell is the result of that. Ester formation, spindle formation, disappearance of nucleolus and formation of equatorial plate. All these things, spindle formation, disappearance of nucleolus and formation of equatorial plate, that forms during cell death. And ester formation takes place only in the animal cell with the help of centriole. Synapses, crossing over, chiasma formation and interkinesis. Synapses, crossing over, chiasma formation, they all occur in the stage of meiotic 1. They all occur in the stage of meiotic 1. Interkinesis is the phase which occurs between the two cell division. Splitting of centromere, formation of daughter chromosomes, reappearance of nucleolus. Splitting of centromere, that occurs during the cell division. Formation of daughter chromosomes is formation of daughter chromosomes that forms during the birth, and reappearance of the nucleolus, that is after the formation of cell. Splitting of cell, formation of daughter, reappearance of nucleus. इसमें जो होगा आपका ऑड वन आउट होगा, स्प्लिटिंग ऑफ सेंट्रोमियर, ये होगा आपका, ये नहीं. Splitting, formation of daughter chromosome and reappearance of nucleolus, both forms at the telophase stage, whereas splitting of centromere, it occurs at the anaphase stage. Centrosome, cell wall, cell membrane, large vacuole. Cell wall, cell membrane, large vacuoles, they are all parts of a cell. Whereas centrosome, it is present in the chromosome from where the centrosome is present the cell wall that occurs with the in the chromosomes, the structure which is found over there. Now the next one, in which state of karyokinesis of mitosis the following events occur? Nuclear envelope disappears, that is during the prophase. Reappearance of nucleolus occurs during telophase. Formation of equatorial plate occurs during metaphase. Formation and movement of astrole towards the opposite pole, it is during the anaphase. Uncoiling and elongation of chromosomes. Uncoiling and elongation of chromosomes in the telophase. Splitting of centromere. Centromere and movement of daughter chromosomes towards opposite poles, that is in the anaphase. Okay, now see the next one. Who coined the term mitosis and meiosis? Mitosis is coined by W. Fleming in 1882. Term meiosis, it is coined by Farmer and Murray in 1905. Name the cell division that takes place during gametogenesis. What is its significance? See, the cell division that takes place during gametogenesis is meiosis. जो गैमेट्स फॉर्मेशन में जो डिवीजन होता है वो होता है meiosis. What is significance? Meiosis is responsible for the formation of sex cells or gametes, and they are responsible for sexual reproduction. And during meiosis 1, at diplotin stage, crossing over takes place, due to which there is a qualitative changes occur. When जब क्रॉसिंग ओवर होती है तो क्रॉसिंग ओवर में मैटरनल और पैटर्नल क्रोमोटिड्स जो होते हैं वो आपस में एक्सचेंज होते हैं, जिससे कि कैरेक्टर्स में एक नया कॉम्बिनेशन बनता है. फिल इन द ब्लैंक्स. पेयरिंग ऑफ होमोलोगस क्रोमोसोम्स इज नोन एज सिनेप्सिस एंड टेक्स प्लेस इन zygote. क्रॉसिंग ओवर इज द म्यूचुअल एक्सचेंज ऑफ करस्पॉन्डिंग पार्ट्स ऑफ नॉन सिस्टर क्रोमोटिड्स, क्योंकि अपोजिट मैटरनल और पैटर्नल के बीच में होता है. आफ्टर क्रॉसिंग ओवर, पॉइंट ऑफ अटैचमेंट बिटवीन नॉन सिस्टर क्रोमोटिड्स, दे आर कॉल्ड किएस्मा. किएस्मा फॉर्मेशन क्रॉसिंग पॉइंट का होता है. meiosis 1 is also known as reduction division. Chromatids are two strands like structures of one chromosome which are attached at a point of centromere, right? Define cell organelle. Cell is the structure and the functional unit of life. Cell organelle, membrane-bound, well-organized protoplasmic structures. They are the parts of a cell which perform various functions. They are called as cell organelle. Mitosis division results in the formation of two daughter cells with same number of chromosomes, right? Meiosis or meiotic meaning is same. Meiotic division results in the formation of gametes where the number of chromosomes becomes half of the parent cell. Evolution, a progressive change in the genetic makeup of a population with accompanying changes in the characteristics of the organism. That is what called evolution. Crossing over, an exchange of corresponding segments of the chromatids of homologous chromosomes occurs during meiosis, right? Karyokinesis, it refers to the division of nucleus diploid having two sets of chromosomes in each cell. And interphase, the stage before beginning of mitosis, is called interphase. In this phase, genetic component duplicates. Next, differentiate mitosis in animal cell and plant cell. Just let us see it over here. Mitosis in plants and mitosis in animal cell. In plant, cell plate is formed. In animals, cleavage is formed. In plants, centrosome absent. In animals, centrosome is present. In plant, formation of asters is not seen. Here, formation of asters and spindle is seen. Here, a cell plate is laid in the cytoplasm at the equatorial plane in telophase. Here, cytoplasm contracts in Telophase, no cell plate formation occurs in case of animal cell. Mitosis and meiosis. See, so there's a lot of differences are there, but I prefer only the one. Mitotic division occurs in the vegetative parts of the cell or somatic cells in case of animal cell, whereas meiotic division occurs in the reproductive cells during gamete formation. जैसे sperm and ova formation in case of the humans, in case of plants, formation of pollen grains and formation of ovule. The reduction division occurs. In mitosis, chromosome numbers remain the same that of the parent cell after meiotic division. Here, number of chromosomes is reduced to half after the meiotic division 1. Next, centrosome and centromere. Centrosome, it is present in the animal cell. Centromere, it is present in the chromosome. Centrosome, it helps in cell division. Centromere, the two chromatids are joined at one another at a point that is called the centromere. That is the connection point between the two chromatids. Cytokinesis and karyokinesis. Cytokinesis, it is the division of cytoplasm, the daughter cells having equal distribution of cytoplasm. That it and its constituent means whatever the cell organelles are there, when the nucleus divide, after the division of the nucleus, other cell organelles are also completely divided into two parts. Constriction occurs and one cell divides into two. Karyokinesis, this is the division of the nucleus into two daughter nuclei of about equal size. When the content of the cytoplasm increases, single nucleus is not able to control, then what happens? Nucleus starts elongating, divides into two as a mitotic division, and then the nucleus separate. Later on, cytoplasmic inclusions, they are also separate. That is the cytokinesis. First karyokinesis occurs, then cytokinesis occurs, and the cell is divided into two parts. Now, how mitosis differs in animal cell and plant cell? We have already discussed over here difference between mitosis in plant cell and animal cell. Here we have already discussed. Now, what is meant by cell cycle? Discuss its various stages. See, cell cycle. Cell cycle is the sequence of metabolic steps during which a cell prepares for and conducts DNA synthesis and mitosis. It consists of G1 phase, S phase, G2 phase and M phase. What is first G1 phase? It is also known as post-mitotic phase. In this phase, synthesis of protein and RNA takes place. The total time taken for completion of this phase of mitotic cycle is 30 to 40%, and the chromosomes are fully uncoiled and thin and form a reticulum. Next comes S phase, phase synthesis of phase of DNA synthesis, means DNA duplicates. Now in this phase it follows G1 phase. मतलब ये G1 phase के बाद आता है. It phase follows G1 phase. DNA and histones are synthesized and each chromosome divides into two chromatids, and the time taken for this phase is 30 to 50%. इसमें 30 to 50% होता है, यहाँ पे 30 to 40% होता है. Next one, G2 phase, that is pre-mitotic. This phase follows S phase. It is the phase for preparation of mitosis, and the time taken for this phase is 10 to 20%. Now the next M phase or mitotic phase. Now here all the growth occurs. Now the nuclei divide centromere as the chromatids, they split. They are duplicated already, growth occurs, and then only they split with the centromere and form the two cells. This phase follows G2 phase and includes prophase, metaphase, anaphase and telophase, and the time taken for this phase only 5 to 10% of the total mitotic cell. All this including together, it is termed as cell cycle, right? Now the next, discuss the significance of mitosis and meiosis. Now what are the significance of mitosis? In this division, the chromosome number in each daughter cell remains the same like the parent cell. Second, it helps in the growth and development of organ and the body of the plant. Third, it helps in replacing the old, decaying and dead cells by the new ones. it helps in the asexual reproduction, and the fifth one, it helps in maintaining the same combination of genes. I hope it is clear. Now the next one, significance of meiosis. Meiotic division helps in restoring the chromosome number, characteristics of the species. Second, it is necessary part of the life cycle of sexually reproducing organism. Third, during meiotic division 1, crossing over takes place which results in recombination of genes. This helps in improving the species. And the fourth one, recombination of genes causes variation in the members of the species, and variations provide raw materials for evolution. Select the most appropriate option. A cell has 5 pairs of chromosome after mitotic. So number of chromosome, see 5 pairs means number of chromosome will be 10. So it will be 10, 5, 10, 20, 40. How many? In daughter cell. And chromosomes mitotic means numbers remains the same. And 5 pairs, and here it is number of chromosome, not chromosomes, number of chromosome. तो number of chromosomes जो होंगे वो होंगे 10. The plant where 2 non-sister chromatids cross over, they are called chiasmata, centromere, chromatids. The where 2 sister chromatids cross over, crossing का point जो होता है वो chiasmata होता है. The point where 2 sister chromatids of a chromosome are attached, that is known as centromere. Through the centromere, they were attached. Chiasmata इसे जो linkage होती है, crossing over का point जो होता है वो chiasmata होता है. और centromeres, the point where the chromatids are attached. Sister chromatids are attached. Which of the following is known as post-mitotic phase? G1 phase, G2 phase, M phase, none of these. G1 phase is known as post-mitotic phase. Which of the following is the reduction division? Meiosis 1, meiosis 2, mitosis, interphase. It is meiosis 1. Explain the structure of chromosomes in eukaryotic chromosomes. Structure of chromosome, as you see, they are the helical structure. Each chromosome consists of 2 chromatids which are joined at a region called centromere. देखिए ये point है centromere जहाँ से ये 2 chromatids joined होते हैं. Each chromatid consists of a number of thread-like chromonema. यानी कि ये जो होते हैं जो 2 threads हैं, ये single 2 thread हैं, ये हैं chromonema. जब ये होता है इसमें जो 2 pairs होते हैं ये break होते हैं और ये अपना पेयर बना लेते हैं, तो यही डुप्लीकेशन होता है क्रोमोसोम्स में. विद ईच अदर दे आर एम्बेडेड इन ए मैट्रिक्स, यानी कि ये जो फ्लूइड है ये मैट्रिक्स कहलाता है. मैट्रिक्स एंक्लोज्ड इन ए शीट ऑफ पेलिकल. ये जो राउंडेड स्ट्रक्चर है इसको हम पेलिकल कहते हैं. ये जो स्ट्रक्चर है ये पेलिकल है. बोथ मैट्रिक्स एंड शीट दे आर नॉन जेनेटिक. मैट्रिक्स और पेलिकल इसका हेरिडिटरी कैरेक्टर से कोई लेना देना नहीं होता है, सिर्फ ये एसेसरी पार्ट्स होते हैं जो इसको प्रोटेक्ट करते हैं. ईच क्रोमोसोम शोज कॉन्स्ट्रिक्शन एरिया एट ए फिक्स प्लेस. ये प्राइमरी कॉन्स्ट्रिक्शन, ये सेकेंडरी, ये प्राइमरी कॉन्स्ट्रिक्शन, ये सेकेंडरी कॉन्स्ट्रिक्शन. 1 or more centromere present in this. The chromosomal ends are known as, ये जो एंड सैटेलाइट बॉडी है, जो एंड्स होते हैं इन्हीं को हम टीलोमियर्स कहते हैं. और ये टीलोमियर्स ये क्रोमोसोम्स को प्रोटेक्ट करता है. At this season, the chromosomal ends are known as telomeres. Sometimes the chromosomes bear knob-like structure known as, जो telomeres होते हैं इन्हीं को ही हम सैटेलाइट कहते हैं. सैटेलाइट ही टीलोमियर कहलाता है. And the function of chromosome is to carry the genetic information from one generation to another. इस क्रोमोसोम से जो हेरिडिटरी इंफॉर्मेशन है हमारी एक जीन से एक जनरेशन से दूसरे जनरेशन. Why is cell division necessary? Now see, cell division plays an important role in all living organisms as it is essential for growth, repair and reproduction. This process helps in renewing of damaged cells, production of new cells from older ones and maintenance of chromosome number. So cell division is necessary. Now in the diagram given represents a represents a certain phenomena occurs during meiosis. See, they are homologous chromosomes, they are chromatids, they are exchanging material. ये यहाँ पर देखिए क्रिस्मा फॉर्मेशन हुआ. अब देखिए ये एक ब्लू है, ये ग्रीन है, इसने क्रॉस किया तो इस जो ब्लू वाले का था ये आधा ये कट के यहाँ चला गया और ग्रीन वाले का आधा कट के इसमें जो है, इसी को हम क्रॉसिंग ओवर कहते हैं. नेम द फिनोमेना शोइंग हियर, इट इज द क्रॉसिंग ओवर. Define crossing over, that is the mutual exchange of corresponding parts of the non-sister chromatids. State the difference between the chromosome and chromatids. See, chromosome at the time of cell division, chromatin coalesce and forms thread-like chromosomes. वो तो क्रोमोसोम्स होते हैं, ये पूरा क्रोमोसोम. एंड ड्यूरिंग द सेल डिवीजन ईच को स्प्लिट्स लॉन्गिट्यूडनल एक्सेप्ट द सेंट्रोमियर, मतलब ये अटैच्ड है सेंट्रोमियर से. तो ये जो पॉइंट होता है सेंट्रोमियर, और जो पार्ट्स हैं ये क्रोमोटिड. सो दे आर नोन एज पार्ट्स ऑफ द स्प्लिटिंग क्रोमोसोम, दे आर नोन एज क्रोमोटिड. जो अटैच्ड होता है ये सेंट्रो, और पूरा इसको क्रोमोसोम बोलते हैं. गिव वन पॉइंट ऑफ डिफरेंस बिटवीन द फॉलोइंग, मेटासेंट्रिक एंड टीलोसेंट्रिक. मेटासेंट्रिक मतलब सेंट्रोमियर लाइज इन द सेंटर ऑफ द. टीलोसेंट्रिक सेंट्रोमियर लाई एट द वन साइड एंड एंटायर ये यहाँ पर होता है. सेंट्रोमियर एंड टीलोमियर लोकेशन, यहाँ सेंट्रोमियर की पोजीशन आपसे पूछिए तो इसके एक साइड में होता है, यहाँ पर सेंटर में होता है. सेंट्रोमियर, द सेंट्रोमियर इज रिक्वायर्ड फॉर सिस्टर क्रोमोटिड्स टू ज्वाइन अंटिल इट बिकम्स इन्वॉल्व इन द क्रोमोसोम सेग्रीगेशन इन ए सेल डिवीजन. इससे क्रोमोसोम स्प्लिट होते हैं. टीलोमियर, it is protects the chromosome ends of the and degradation of the fusion of the and present chromosome shortening during DNA. जब DNA replicate होते हैं, shortening chromosomes होता है, तब इनके ends को ये protect करता है. Maternal means the mother prepared the chromosome, means ovum 23 paired, means from the mother side is ovum, that is called the maternal sex chromosome, and father prepared each sperm with the 23rd 3 chromosomes, that is called parental chromosome, that is sperms formation. Sperm, it is the paternal chromosome, ovum, it is the maternal chromosome. Allosome means sex chromosome. Idiosomes या sex chromosome या allosome तीनों एक ही चीज़ें हैं. Chromosomes controlling sex of the individual, they are sex chromosome. Other than sex, other chromosome मतलब in the human 23 pairs are there, तो 22 pairs they are autosomes. And the 23rd pair only, single one, this is known as sex chromosome. मतलब सेक्स जो डिटरमाइन करता है सेक्स के, जैसे हमारे हाथ हैं, पैर हैं, बॉडी ऑर्गन्स हैं, ये सब जो हैं, ये सारे जो क्रोमोसोम करते हैं वो ऑटोसोम्स के जरिए करते हैं. एक्सेप्ट सेक्स क्रोमोसोम, ऑल अदर क्रोमोसोम्स दे आर नोन एज autosomes, right? Now the diagram given below you can see represent stage. Name the stage. It is anaphase, second of the meiosis. Is it a plant cell or animal cell? Give it the reason. Here as the cell plate is seeing, so it is a plant cell. In plant cell, formation of cell plate is in the middle, so it is there. Label the parts. देखिए ये centromere है, ये chromatids है, ये cell plate है, ये spindle formation है. Draw a well-labeled diagram metaphase. In metaphase, all the chromosomes are lying at the center. Centromere is present in, this is the centromere. They are the chromatids, these are the chromatids, and this is the equatorial plate, and these are spindle formation. Now given below a diagram representing a stage during mitotic cell division in an cell. Now ये identify the above stage given a reason in support of your answer. See this, as here you see nuclear membrane disappearing, means it is the late prophase. It is showing disappearance of nuclear membrane, means it is a late prophase. And name the cell organ that forms the astral, that is the centrosome. जो इसमें ये astral rays जो होते हैं, astral body के द्वारा astral rays form होती हैं जो कि centrosome में present होती हैं. Now here name the parts labeled centromere, chromatid, spindle. Name the stage that follows the stage of mitosis. How can this stage be identified? See, before the late prophase, it is the middle prophase, not this one. It follows middle prophase. Middle prophase stage. where the disappearance of nucleolus occur. Here the disappearance, what happens? Middle prophase, how can identify? Disappearance of the nucleolus, and in the late prophase, disappearance of the nuclear membrane occurs. Mention 2 points of difference between mitosis and meiosis. See, number out. In mitosis, 2 daughter cells are formed. In meiosis, 4 daughter cells are formed. In mitosis, chromosome numbers remain same in the daughter cell. In meiosis, chromosome number is reduced to half of the parent cell. Now, karyokinesis and cytokinesis. Explain the term. We have the division of nucleus is called karyokinesis, division of cytoplasm is called mitosis. In number of daughter cells, in mitosis 2 daughter cells are formed. In meiosis 4 daughter cells are formed. Tonoplast and plasma membrane. Tonoplast, it is the outer covering of the vacuole. Plasma membrane, outer covering of the cell membrane, that is called the cell membrane. Why gametes have a haploid number of chromosomes? Gametes have a haploid number of chromosomes so that when the male and the female gametes unite, then they form the same number of chromosomes as that of the parent. They have haploid, so they maintain the number of chromosomes in the coming generation and in the next generation. I hope everything is clear. Any doubt anywhere, you can ask me in the comment section. It's part 2. I will give you in the next video. Some questions are still left over that I will discuss in our next video. Thank you. Again, we will meet in the next video.